So I'm James from Refeyn, and we are in Amsterdam at the moment at the BPI conference, and we are exhibiting our products and our technologies to the bioprocessing community.
What does mass photometry do for the bioprocessing community?
So it's a good question. Mass photometry is it's a brand new analytical method. When I say brand new, we launched in 2018, so it's a few years old now, but obviously with a couple of years of awkward situations over the last few years. But we're very proud. We've sold over 200 instruments in the field, and it adds value in a number of areas. So it's an analytical method to look at single particles from individual proteins all the way to small viruses. And as a single molecule method, it really adds a much higher resolution than you would get on traditional analytics, which use batch methods. Specifically for the bioprocessing community, we can work with antibodies really nicely. Our mass range goes down to 30 kilodaltons all the way up to about 5 megadaltons. So looking at antibodies, antibody stability, antibody binding to antigen, we can look at all of these things. We also get good readouts on antibody aggregation, all on our 2MP platform. And then going towards small viruses, we can also start working towards there as well. So it adds a brand new analytical method. It's extremely quick and easy to use. Measurements can take approximately about a minute. The barrier to entry is really, really low, so it can be used by anyone in the company. And as a benchtop piece of kit, you get the access to your data extremely rapidly to allow you to design your next experiments and purifications downstream.
How is this advancing the field of biomolecular analysis?
So I think one of the key aspects which I touched on is that speed and ease of use. I think there's a lot of great analytical methods out there, and we're not trying to replace any of them. We're a firm believer in all of these complementary methods. Where our platform is used quite successfully, because it's benchtop, it's in the same lab that people are using their other instruments for. They're producing these molecules, and as the measurements can take only a couple of minutes, you can get your data really, really quickly. And allow you to then impact on your next experiments within the next hour. There's some other methods that give extremely rich data but might take hours or even longer to make the measurements. Our method then allows you to try perhaps more conditions in a short period of time and using very, very little sample. I think this is key to a lot of bioprocesses these days. A lot of these molecules are very difficult to produce in any significant amount. And if you have to use 50 or 60% of that production run for analytics, you get stuck in a loop quite quickly. So mass photometry has the advantage of using very, very little sample.
What should scientists who work with AAVs know about this method?
So the AAVs is a newer market to us, let's say, but as I said, we only go back to 2018, 2019 anyway. Mass photometry was used by some people on our old system to look at AAVs. Since then, we've launched a product line called the SAMUX, which is a mass photometer optimized for AAV measurements. What this allows us to do is to very rapidly determine the empty-full ratio of these AAV capsids. So one of the big challenges in the field is to determine post-production what you actually have. The active drug in the AAVs is the nucleic acid cargo. And if you don't know how much of your virus has that cargo in or how much is empty, you really don't know what you're producing. So there's the need for a very rapid analytical method to determine these accurately. At the moment, the two other methods that are extremely popular to measure these things are electron microscopy or analytical ultracentrifugation, AUC. Both great methods, where again we have a slight difference is we're a lot quicker, we're on the bench to do these things. So for AAVs, our SAMUX product is perfectly suited to measure the empty-full ratios, again, in a couple of minutes on the bench.
And is this method suitable for automation?
Yes, good question. So this is something we looked at a long time ago. A lot of our early pharmaceutical customers, of course, wanted some higher throughput, some more automation. This was something that was always on the cards. About 18 months ago, we launched an automated system for our 2MP. So this is the flagship product that goes down to about 30 kilodaltons, can measure antibodies, other biomolecules. The Samex Auto we also have launched as well. So yes, we have an automatic platform for it. It allows the automatic measurement of up to 24 samples from a 96 well plate format. It increases reproducibility because you now have a robot doing the pipetting rather than a human who's had perhaps one or two few coffees than they should have had. And allows a walk away time of 90 minutes or so, whereas before, one of the drawbacks of our system was it was too quick. If the measurements take a minute, you don't have time to get your gloves off before you want to make the next one. Now we can load the system up with up to 24 samples and go and do something more useful while that automatically runs. Data is automatically processed.
Okay. And when do you think we can expect to see this used within a GMP setting?
So GMP is something that, again, was a natural evolution from us, from the markets we started selling these systems into. Very soon is the good news. So hopefully later this year, we will have GMP compliant software. That will be rolled out as an upgrade to existing customers if they wish it, and also then part of the package going forward for new systems. So the instrument fundamentally is fine. We just need to get the software sorted, and that will be done later this year.
And if someone was interested to find out more, how can they find out about mass photometry?
So our website is obviously first port of call. You can get in contact from us for our website, get direct contact with them with our team of application specialists. We have a team, large team in Europe. We're an Oxford-based spin-out, but we also have offices in Portland, Oregon and Boston, as well as a small company in Japan as well. So our website is full of information. Get in contact with us if you need more. We also host a lot of events. So later in May, I believe May 23rd, we're hosting a user meeting in our offices in Boston. So people are welcome to register for that. We will have our existing instruments, some sneak previews of our new instruments coming there as well for people to see.
Exciting.
So yeah, a lot of information out there, but the best thing is really just get in touch and then we can have a conversation, work out what people's needs are and how we can help.
Is there a particular theme or hot topic that everyone in the industry is talking about this year?
I think one of the topics that's almost always there is on the reproducibility front. And I think especially when it comes to a lot of cell and gene therapy and new vectors, we're at a relatively early stage of drug discovery compared to traditional small molecule discovery. And that need for reproducibility in terms of production, titer, optimizing all of these processes along the way, there's an awful lot of effort going into there. So I think any tools that can aid that journey are really needed.
And why do you attend events and conferences? What do you find the benefits are?
I think it's multifactorial really. It's a great opportunity for us to get out and see some of our customers. We do a lot of this anyway. We've obviously had a couple of years where that's been difficult. Conferences are a great one-stop shop. We have a lot of customers here. We also have a lot of people who we've spoken to in the past, but getting a face-to-face meeting with them is so much better. So it's a great opportunity to see a lot of people in one place. It's also a great opportunity for us to work with other companies who might be exhibiting and see what they are doing. So yeah, ticks a lot of boxes for us.
Great, thanks.